Good afternoon to all the students and welcome to NPTEL problem solving session. We'll be solving week 10 week 10 practice problem set corresponding to the lectures of week 10 where Professor Subramaniam in the course Introduction to Developmental Biology is talking about mammalian development as well as plant development. So to all the students who have attended the previous sessions and to the new students, the same set of general rules remain. Please turn off your mic and video chat during the session for smooth conduction of the session. And this will be an interactive session where we will solve example questions corresponding to the current lecture week, which is week 10. We'll solve a set of 10 to 11 questions, multiple choice, similar format to what we expect in the quiz and in the exams, which will be covering all the topics corresponding to the current lecture series. And then I'll ask you guys to post the correct answer in the chat box. So all the interaction will be via the chat box. You guys will post. I would ask you guys to post the answer in the chat box, and then after collecting some of the answers, I'll reveal what is the correct answer and we'll understand the rationale behind why the correct answer is the correct answer. So since we'll be dealing with plant developmental and animal development post-fertilization, today in the practice problem dealing with lecture week 10 by Professor Subramanian. So we'll start shortly. I'll present my screen at 3:05. Meanwhile, I'll keep admitting students who are requesting to join this call, and hopefully this will be a fruitful session for all of us guys where we'll have a two-way interaction while solving these questions together. We'll start soon at 3:05. Okay students, as we can see, it's already 5 minutes past 3. We'll be starting the problem solving session right now. What I'll do is I'll present my screen and switch on my laser pointer and ask you guys for confirmation for audio and slide check. Let me just quickly present my screen. I hope you can see my screen. I hope you can see my screen and can hear me properly, and also the slides changing. Just let me know with a yes or no, and then we'll proceed forward. Okay, cool. So once again, I welcome you all to the problem solving session, practice assignment week 10, Introduction to Developmental Biology. And the instructor for this course is Professor Subramaniam. My name is Ashutosh and I'm a PhD scholar at IIT Kanpur. So the way we'll be proceeding through this problem solving session is I'll keep posting relevant questions corresponding to week 10, and then we'll try solving for the correct answers. I'll reveal the correct answer and then we can move on to the explanation of the correct answer. Cool. And during this session again, I would request to turn off your mic and video chat and interact with me via the chat box. So the first question of the session: which of the following statements are false? This is the first question. Which of the following statements are false? Option A: apical basal polarity is clearly visible in the heart-shaped embryo of plants. Option B: meristem in plants is similar to the stem cells in animals. Option C: the endosperm initiates photosynthesis and stores food. Option D: the shoot and root meristem are formed independent of each other. Which of the following statements are false? Okay, we have one answer saying the last, shoot and root meristem form independent of each other. Let us check what is the correct answer. The correct answer is option C, endosperm initiate photosynthesis and stores food. This is a false statement. All other statements are true statements. Apical basal polarity, heart-shaped embryo is true. Meristem is similar to stem cells is also true. And shoot root meristem formed independent of each other. Now if you look at the heart stage embryo of the plants, we can appreciate that the outer layer, this outer layer is the covering of the cotyledon. We have the shoot meristem at the tip forming the apical side, and the root meristem forming the basal side. So here we can distinguish between shoot and root meristem, and we have the procambium, which will further lead to lateral meristems, or in the girth, increase in the girth of the plant. So we have the establishment of root shoot axis in the heart stage of the embryo itself, and we can say that shoot meristem is towards the apical side, root meristem is towards the basal side, and that's why axial patterning is clearly evident in the heart stage of the plant embryo. While in the globular stage, we can say that radial patterning is evident. Now on the second point about meristems, plant stem cells versus animal stem cell, here's the comparison between the meristems and the animal stem cells. Undifferentiated cells located in the meristem part of the plant. So we see that plant stem cells are also called meristem. Cells that produce new cells for the regeneration and repair of the existing tissues are stem cells in animals. Plant stem cells occur in meristem, whereas animal stem cells occur in most animal tissues. So we have what we call tissue resident stem cells, wherein each organ and almost all organs have their own kind of stem cells which can help them in repair and regeneration. Plant stem cell or meristem are responsible for continuous growth of the plant body, whereas animal stem cells are mainly responsible for regeneration and repair. Located at the terminal of the inner hair cells, located at the terminals of the outer hair cell nerves. So these are another location based difference, are totipotent and are multipotent. So the meristem, the stem cells that we have in the meristem of the plant are totipotent. So they have the capability of giving rise to the whole plant itself. But in animal stem cells, we have stem cells with lesser potency or multipotent, which can give rise to a fewer cell types corresponding to the particular organ it is residing in. So this is a comparison between meristems, which are plant stem cells, versus animal stem cells. And if we see for the last option, where we have a comparison between shoot apical meristem and root apical meristem. So this is what I was talking about. So if we look at the terminal bud in the plant and the axillary bud, the terminal bud contains the shoot apical meristem, and a cross-sectional view would look like this. This part is called the shoot apical meristem. Then we have leaf primordia, from where the new leaves grow out. And in woody plants, the vascular cambium and cork cambium thicken the stem and root. So these tissues arise from the procambium, which then form the vascular cambium and the cork cambium, which can increase the thickness of the plant. These are called lateral meristem, lateral growth of the plant. Then at the basal side, we have root cap, or which guards the root apical meristem. So here is a representation cross-section of root cap with root apical meristem. While both shoot apical meristem, SAM, and root apical meristem, RAM, are essential for plant growth and development, they have different functions and develop independently of each other. So they are developing independent of each other. The growth and differentiation of cells in SAM and RAM are regulated by distinct set of genes and hormones to ensure the proper development of both shoot and root systems in plants. So not the single set of genes is responsible for both SAM and RAM. So there are distinct set of genes and both can develop independently of each other. That is why the false statement that endosperm initiates photosynthesis and stores food, this statement is false, because endosperm just stores food, but initiation of photosynthesis is done by leaves of the plant or the cotyledons. When the cotyledon arises, then we have the first leafing primordia, and we see the initiation of photosynthesis. So endosperm stores food was correct, but initiation was false. And we just saw the other three statements. Heart stage, axial patterning, plant stem cells are also known as meristem, and both SAM and RAM develop independently of each other with different gene set controlling their development. I hope this is clear to the students. With a yes or no, just let me know. Then we'll move on to the next question. Yes, I understood that. Okay, thank you. Let's move on to the next question. This is a match the following question. We have 1, 2, 3, 4 matching with A, B, C, D. Now you have to find out which is the correct match. On the left hand side, we have radical, abscisic acid, seed coat, procambium. On the right hand side, we have dormancy, scarification, vascular tissues, imbibition. And the options here are 1A, 1A, 2C, 3B, 4D. The next option 1B, 2C, 3D, 4B. The third option 1D, 2A, 3B, 4C. And the last option 1B, 2A, 3D and 4C. So these are the four matches in the options. Now it's up to the students to answer what do they think is the correct answer, the correct matched option. We have radical, abscisic acid, seed coat, procambium. On the right hand side we have dormancy, scarification, vascular tissues and imbibition. Let's take a minute to collect some answers and I'll reveal the correct answer. So I see there is a answer. Ajay is mentioning option 3. So let us see if what is the correct answer for this question. And you are absolutely correct. Option C is the correct match. So radical matches with imbibition, abscisic acid with dormancy, seed coat matches scarification, procambium matches vascular tissues. So let us look at these processes one by one. Seed germination process. In the process of seed germination, imbibition is the process of water uptake by the tissue. Water uptake softens inner tissues, causes swelling and seed coat rupture, more water uptake. So this is the process of imbibition. It's kind of a chain reaction. So the first initiation, small water uptake, soften the inner tissues, which causes swelling and seed coat rupture. Once the seed coat is ruptured, then we have more water intake. And here is an example of barley seed germination, where the radical or the future root, will be the future root and the plumule will be the future shoot. So the radical will help in water imbibition, which will soften the inner tissues, swelling, seed coat rupture, and then more water can uptake. So in other parts of the seeds are labeled cotyledon, epicotyle, radical, and this is the aleuron cells and the endosperm storing the starch. So once the seed coat is out, then we can have the cotyledons blooming and then the seed germination. So the process of seed germination started by imbibition of water through the radical. Now the second matching column, we had abscisic acid. Abscisic acid is abbreviated as ABA. ABA is the main reason for seed dormancy. So seed dormancy is a phenomenon in which a seed does not germinate until and unless it finds certain chemical cues, and when it senses the environment is right for its germination, for its survival, only then the seed will germinate. And dormancy is in a state where metabolic activity of the seed is also ceased and it is in a resting state. So abscisic acid, a hormone, plant hormone, is the main reason for seed dormancy. Abscisic acid inhibits seed germination. Actually, it is the ratio of abscisic acid to gibberellic acid. Gibberellic acid is another hormone in seeds that determines whether the seed remains dormant or germinates. So the ratio to ABA to GA will determine whether the seed will remain dormant or whether it will germinate. And here is a representation structure of abscisic acid. So it's an aromatic compound. We have a cyclohexenyl ring and many other groups, functional groups. So abscisic acid is the main reason for seed dormancy. And the third massive column was about procambium. Procambium is a primary meristematic tissue found in the plants. and it plays a crucial role in the development of vascular tissues, which are responsible for the transport of water, nutrients, and food throughout the plant. So the procambium, as I was talking about, will form later the lateral meristems, and it also gives rise to the xylem and phloem of the plant, which are two major pipelines through which water and food are transported throughout the plant respectively. Xylem transports food and phloem transports xylem transports water and phloem transports food inside the plant. Role of procambium in vascular tissue development: it is a precursor tissue for both xylem and phloem. Procambium cells can differentiate into tracheids and vessel elements to form the xylem, and sieve elements and companion cells for the phloem. So these are special differentiated cells that form the specific tubes of xylem and phloem. For food transport, we have phloem, where we have sieve elements and companion cells. Food, I mean here, is starch, and tracheids and vessel elements for water transport, columnar water transport by xylem. So if you cut a cross section through the girth of a plant, we can see this thin tissue lining is the procambium. This center part is a pith. Procambium gives rise to the blue part, which is the phloem, and the pink part, which is the xylem. So it gives rise to two differentiated tissues which have immense function in the plant. So that's why we have three options matching: radical to imbibition, abscisic acid, dormancy, procambium, vascular tissues, and seed coat will be scarification. I hope it is clear to the students. Okay, let's move on to the third question. Choose the correct option. We have two statements here and we have to check which of these is the correct option. Plant chimeras can have variegated leaves if the L2 apical meristem of a plant lacking chlorophyll is grafted onto a plant producing chlorophyll. So this is a tricky statement. Plant chimera, so we have a plant chimera, and the question is whether it can have a variegated leaf. Variegated leaf, mix patched leaf, or patched colored leaves, not totally green, if the L2 apical meristem of a plant lacking chlorophyll. So what we do, we graft the L2 apical meristem of a plant lacking chlorophyll is grafted onto the plant producing chlorophyll. So we have another plant producing chlorophyll. We cut out the L2 apical meristem of the first plant which lacks chlorophyll. So we graft that L2 onto the plant producing chlorophyll. Then can we have a plant chimera which can have variegated leaves or multicolored leaves. Here multicolored means leaves with patches of green and colorless, because the L2 plant is lacking chlorophyll. And statement number 2, different layers of the shoot apical meristem L1, L2, L3 give rise to different structures. So this is a question about layers of the shoot apical meristem and a transplantation experiment. And the options are: statements 1 and 2 are true. Statement 2 is the correct explanation of statement 1. Option B, statements 1 and 2 are true, but statement 2 is not the correct explanation of the statement. Option C, statement 1 is true, statement 2 is false. Option D, statement 1 is false, statement 2 is true. And option E, statements 1 and 2 both are false. So here are two statements and these are the options. Now let's take a minute to collect some answers. I can see Ajay has already answered, but we'll wait for some time and let other students wonder about the question. So you need to have a basic understanding of the shoot apical meristem to answer this question. It will be easier for you guys. Okay, let me reveal the correct answer to this question. The correct answer is option 1. Statements 1 and 2 are true, and statement 2 is the correct explanation of statement 1. And Ajay is absolutely correct. Well done. So let us have a look at the layers of the shoot apical meristem and understand a bit. So if you look at the shoot apical meristem, it somewhat looks like this. We have two leaf primordia as we saw in the first figure, and this is the shoot apical meristem. Now within the shoot apical meristem, we can divide it into various zones. The first prominent zone is the central zone marked in green. The central zone of SAM is the shoot apical meristem, contains most undifferentiated and slow dividing cells. These cells serve as stem cells. So central zone has slow dividing cells or stem cell, which are not usually committed to a particular fate, let's say leaf or flower or stem. Cells in this zone give rise to daughter cells that eventually differentiate into various tissues of the shoot, including leaves, stems, and flowers, as I talked about. So later on, these stem cells will undergo differentiation to give rise to leaves, stems, and flowers. Then we have peripheral zone, these red zones on the periphery of the shoot apical meristem. Surrounding the central zone, the peripheral zone consists of cells that are more actively dividing and differentiating. So here the cells are more actively dividing and differentiating away from the stem cell niche like we have in the animals. The cells in this zone contribute formation of the stem and leaf primordia. So the leaf primordia that we see here, cells from the peripheral zone form the leaf primordia and also constitute the stem of the plant. Then we have a big orange zone here called the rib zone. The rib zone is located just beneath the peripheral zone. Just beneath the peripheral zone, there is the rib zone. Cells in this region are responsible for producing vascular tissues, xylem and phloem. So the procambium, as we talked about earlier, procambium forms from the rib zone, and the procambium has similar properties of the rib zone and produces xylem and phloem. Vascular tissues are essential for transport of water, nutrients, and food throughout the plant. So rib zone produces vascular tissues. Then we have three layers called lateral layers, L1, L2, and L3. The outermost layer of the SAM is called tunica, which is usually 1 to several cell layers thick, the L1 or the yellow layer. The inner layers, collectively known as the corpus, include L2 and L3. So the pink and gray one are the L2 and L3. The tunica provides protection to the meristem. Corpus contributes to the formation of various above ground structures. So corpus will form the shoot structures and the tunica will provide protection to the shoot apical meristem. Just like we have root cap providing protection to the root apical meristem. The L1 layer can give rise to epidermis and some leaf tissues, while L2 and L3 layers produce internal stem and leaf tissues. So L1 can give rise to epidermis and leaves, and L2, L3 for more internal structures. So pink and gray, more internal structures, yellow for protection, and epidermis and leaf. So this is how briefly a shoot apical meristem looks like. Now if you see, this is what a magnified image looks like. L1, L2 and L3, the three layers. We have peripheral zone here, the rib zone or medullary zone, and this is the central zone that I was talking about. So what we do, we graft. In grafting a portion of one of the plant, the scion is joined to another plant, the rootstock. So what we do, when an L2 apical meristem lacking chlorophyll is used as a scion and is grafted onto a chlorophyll producing plant, the two plant tissues fuse and grow together. So once we graft the L2 lacking chlorophyll onto the plant which is having chlorophyll, they will both fuse and grow together and they will form what is known as a chimera. The chimera that results from this grafting contains a mix of cells from both the scion and the rootstock. That means both from the non-chlorophyllated plant as well as from the chlorophyllated plant. The L2 apical meristem of the scion will continue to produce cells that make up the central tissues of the stem, which do not typically contain chlorophyll. So this will make the L21, the L2 meristem will form the internal structures which do not typically contain chlorophyll. And variegation will have variegation because the chimera contains both chlorophyll producing and non-chlorophyllous cells. The leaves that develop on the chimera can exhibit variegated patterns, means mosaic patterns. The parts of the leaf derived from the non-chlorophyrous scion may lack chlorophyll and appear white or yellow, while the parts of the leaf derived from the chlorophyll producing rootstock will be green. So we'll have a patchy appearance with white, green, yellow, green patches. The one which is arising from the transplanted L2 of the apical meristem will appear yellow and white, while the one that is yellow and white, while those tissues which are derived from the chlorophyll producing shoot apical meristem, that will be green in color. So we'll have variegated leaves. I hope it is clear to the students. We'll move on to the next question. Which of the following statements are correct? This is again a true-false statement. Which of the following statements are correct? Option A, Wuschel keeps the cells of the meristem in an undifferentiated state and is negatively regulated by clavata. Wuschel is responsible for cell differentiation and is positively regulated by clavata. TFL1 or the terminal flower gene promotes conversion of vegetative meristem to floral meristem. And D, region-specifying genes like Superman specify the boundaries for the expression of organ identity genes. So these are the 4 statements. You have to tell me what do you think are the correct statements. 2 statements about Wuschel and clavata relationship, one about the terminal flower gene, and another for Superman gene. So 2 about flower primordia, and we have two questions about Wuschel. So let's take a minute and wait for students to look at the question and arrive at the answer. I can see Ajay has already answered, but we still wait for a minute. Okay, it's time for me to reveal the correct answer. The first correct statement, statement number 1, Wuschel keeps the cells of the meristem in an undifferentiated state and is negatively regulated by clavata. This is the correct statement. And the second statement, which is correct, is statement number 4, region specifying genes like Superman specify the boundaries for the expression of organ identity genes. Yes? Ajay was saying something.
I answered same.
Yes, Ajay, I saw it. I was just waiting, and for all those who will watch the video in the future, will have some time to look at the question. I know you have answered, and you have answered very correctly. Good. So these are options 1 and option 4 are the correct statements. Now let us understand the relationship between Wusch and clavata. So this is again a schematic diagram representing the shoot apical meristem with one leaf primordia, and we'll see what happens between these 2 important players, Wusch and clavata. So if you remember, we had an organizing center in the previous diagram where I saw the blue region or the organizing center is the center for the Wusch gene or Wuschel gene, and in the central zone we have the clavata 3. So Wusch is an abbreviation for Wuschel gene in plants that play a crucial role in stem cell maintenance and development. So the organizing center is the center where the stem cells are maintained. So it provides signals which maintain the stem cells in an undifferentiated state. Wuschel is involved in the formation of shoot apical meristem, which is a group of undifferentiated cells at the tip of a plant that gives rise to various above ground plant structures such as leaves, stems, and flowers. So maintaining this central zone or the shoot apical meristem is the functionality of the organizing center by secreting this gene, and this gene product Wuschel. Mutations in the Wuschel gene can lead to abnormal growth patterns in plants because then the shoot apical meristem maintenance will be down. Clavata likely refers to the clavata genes which are involved in regulating size and maintenance of the stem cell population in the shoot apical meristem. Clavata genes interact with Wuschel to control the balance between stem cell proliferation and differentiation in plants. Wusch is a pro-proliferative gene, clavata is a pro-differentiative gene. So Wusch does stem cell promoting transcription factor, maintains the stem cell. Clavata 3 is a differentiation promoting peptide, and clavata 3 inhibits Wusch and promotes differentiation. So Wuschels keep the cells of the meristem in an undifferentiated state and negatively regulated by clavata. So this statement is correct. And if you see about the other two statements that we have, TFL1, we encountered this with TFL1 or the terminal flower gene 1. TFL1 is known for its role in regulating the transition from vegetative phase to reproductive phase in plants. Reproductive phase while production of the flowers and the androecium and the gynoecium. In other words, it controls when a plant starts producing flowers. So this is what I was talking about. TFL1 accomplishes this by inhibiting the activity of genes that promote flowering such as FT, flowering locus T. So we have leafy, we have AP1 cal. So leafy, AP1, these genes promote flower formation, while TFL1 will inhibit, are mutually inhibited. So these relationships are mutually inhibitory. TFL1 will promote shoot formation and inhibits the flower formation. TFL1 actively inhibits AP1 and leafy, which is responsible for flower formation. So the statement would have been correct if it was TFL1 actually promotes more vegetative phase rather than the reproductive phase, which was the opposite statement that we encountered here. TFL1 promotes conversion of vegetative meristem to floral meristem, which is not true. It rather favors a vegetative state than the floral state. And the last statement, Superman. Superman is another gene which acts as a transcriptional repressor controlling the expression of other genes involved in floral development. It is particularly important for proper development of flowers' central region. So Superman is a transcriptional repressor, the expression of which will determine the organ boundaries and the size of the organs. So here is a mutant of Superman where the floral margin is enlarged. So soup generally what it does, it inhibits YUC1/4. This is a protein which enhances the action of auxin along with its partner. When we have a SUP, Superman mutant, this YUC1/4 is not inhibited anymore and it can signal to auxin. Now auxin can increase the size of the floral. So we have delayed FM termination. So FM is large. FM stands for the floral margin. So we have larger floral primordia now due to mutation in the soup. So Superman acts as a transcriptional repressor, controlling the size of or boundaries of expression of other genes which define the organs. Here we saw flower defining organ or the size of the flower primordia. So we have statement 1 and statement 4 as the correct statement, and Ajay has answered correctly as per other students also. Okay. Let's move on to the next question. Which of the following are wrongly matched? Again, a match the following question. Which of the following are wrongly matched? Option A, Hobbit mutant, improper shoot meristem. Option B, Clavata mutant, extra organs produced. Option C, A petala mutant, missing sepals and petals. Option D, Constance activates flowering locus T. These are the 4 matches, and you have to identify which one is the wrongly matched pair. Let's take a minute to find out the correct answer for all those students who are watching this class. Please try to answer. Which of the following are wrongly matched? Okay, let me reveal the correct answer to this question. The correct answer to this question is option 1, Hobbit mutant, improper shoot meristem. This is the correct answer to this question. I think, yeah, this is the correct answer to this question. Hobbit mutant, improper shoot meristem is a wrongly matched pair. Clavata mutant, extra organs is correct. Apetala mutant, missing sepals and petals is correct. And Constance activates flowering locusti is also correct. Let us look at the explanation. Hobbit is a gene required for root meristem but not for shoot meristem formation. There are some genes which are involved in root shoot formation, and Hobbit is one such gene which is required only for root meristem but not for shoot meristem formation. We have Hobbit gene, inhibition of repressor of auxin response, but Hobbit activates auxin-induced root meristem development. Hobbit is a protein that inhibits another protein that stops the gene expression of genes that auxin causes to be made. So auxin downstream genes are repressed by a Hobbit protein, are repressed by a repressor which is inhibited by the Hobbit protein. So indirectly, it repressor of auxin response. Hobbit inhibits the repressor of auxin response in the shoot, but it activates auxin-induced root meristem development. That's why Hobbit and improper shoot meristem development is wrong matched, but correct match would have been Hobbit mutant, improper root meristem. Let us have a look at the Clavata mutant. In a normal plant, Clavata gene helps control the balance between stem cell proliferation, differentiation in the shoot apical meristem. Just in the previous question we saw Clavata is there in the central zone, whereas Wuschel is there in the organizing center. And Clavata genes are pro-differentiative and they maintain a balance between stem cell proliferation and differentiation in the SAM or shoot apical meristem. When these genes function correctly, they prevent the over-proliferation of the stem cells. So job of Clavata is to keep the stem cells proliferation in check, ensuring that meristem maintains its appropriate size and structure. The balance is crucial for proper formation of leaves, stems, and flowers in the plant. But what happens in some Clavata mutants, there is an excessive proliferation of the stem cells in the shoot apical meristem, which is obvious because Clavata is the one which is restricting or inhibiting the Wuschel gene in the organizing center. So if Wuschel is unchecked, then there will be more proliferation, excessive proliferation. This can result in the formation of extra floral organs, abnormal shoot structures, or other growth abnormalities. So Clavata mutants can have extra organs produced due to excessive proliferation of the stem cells due to overactivation of Wusch gene, which is not in control due to mutation in the Clavata. So Clavata and
One doubt. How will we
yes,
identify extra branch in a plant? For a human being, suppose there is an extra finger, so we can say this is an extra digit.
yeah,
What about plant? Can you say this plant should have 10 branches And these extra branches is because of clavata mutant, 11th branch or 9th branch. Means extra organ is produced, means what is produced? Means instead of stem, is it producing root there, or instead of leaf, it is producing flower there? How will you?
No, is responsible for the shoot apical meristem. So shoot structures like,
Shoot structure, there is an internodal difference, and at every node,
yeah,
it is not specified you will produce 2 branches or 3 branches. How?
okay.
What do you mean by extra organ? I'm not getting that. Extra organ for me, human being, for animals, it is decided. For what plant, nobody says that this plant has got extra branch. I
Actually,
did not
the
heard
branch,
that.
the
Branch
the
with what is meaning of that extra organ? Plant don't have organ, na?
organ here means the leaves, stems, and these
Ah.
structures are referred to as the
So
organs,
is
which
it
arise
not
from
decided?
the stem cells.
Is it that okay, instead of 5 petals, it will produce 6 petals?
Not petals actually, but if you see the branches that you are mentioning, the branch, one branch can have multiple branches due to the not due to the shoot apical meristem, but the axillary bud can have the meristem. But the single shoot apical meristem, which is at the apex of the plant, at a given time, it produces a fixed number of structures. Let's say 2 leaves or certain number of flowers. So that over proliferation of the stem cell will lead to production of more organs from the single shoot apical meristem.
Any examples, sir? That's what. Okay, sir.
Example,
I'm not getting it. Sorry, but it's surprised to see there is an organ in the plant and that too extra. It's for me, extra is good for the plants. It is not good for the humans.
yeah, we try to get mutations where we can have our own benefits. Wait, let me just recall if there are any clavata mutants. I can show you. See, I'll just to clear your doubt. I'll just pitch in this and I can show you something. See, you can see here.
White flower has got 6 part.
Yeah, so
Instead of
from one inflorescence or from one inflorescence we have a fixed number of flowers, and in a clavata 3 mutant at the apical shoot apical meristem we have increased number of flowers with the same characteristic with the 6 number of petals, but now the flower buds, the buds that arise, have increased. So it's, yeah, in a way, you said, as you said, it's a beneficial phenotype to the man if a plant is having such a mutation, if the flower is really necessary to us. But it's a mutation in the shoot apical meristem regulating genes like clavata, where this is, let's say, so it depends on the plant to plant what is the fixed number of structure a single shoot apical meristem can give rise to at a given time point. So misbalance in the clavata or wush gene can lead to such abnormalities.
can we have some other name instead of organ there?
Organ is a structure which is like cell, which has a particular function and is connected to the main body axis, is what I would say.
Plant, I don't think organ is a proper word. If something else is given, it will specify the things. Multiple or multiplicity or like that.
Yeah,
It's more
yeah.
flower, not more organ. No, organ flower cannot be organ.
Flower is considered as a sex organ of the plant or the reproductive organ. You can say that.
Not sex organ. It is the part, reproductive part, gametes part, whatever there. It is what we call is an analog, means ovary, sperms, but actually these are not. They are part of the plant. I'm not any good about plant. Not never interested in pot. But for this, I think I have to mug up this because these are very difficult things.
Yeah,
Developmental biology. These are
no, you can have a relationship inside your mind. We have few important genes like clavata, whoosh, and everything, and then you can kind of predict what could happen if things go haywire. Let's say instead of a clavata mutant, we have a whoosh mutant. So the stem cell pool now at the apical meristem will not be maintained. So there will be lesser progenitor pool from where the differentiated structure can arise. So you can imagine the plant growth will be stunted if there is a
the
mutation in the
terminal
Wuschel
terminal
gene.
flower, branch flower. That's very easy to understand. If terminal is absent, terminal flower will be absent. If branch flower is, gene is absent, it's very good.
Yeah,
These are
yeah.
it's- because for the people who are studying it is as a part time, it becomes difficult. For full timers, it's okay.
Okay, let's move on with the session. Yeah, and as we were seeing, Apetala 1. So if you remember, we had a mutant, Apetala mutant missing sepals and petals. Apetala 1 mutations in the AP1 gene can lead to conversion of sepals into carpels, loss of petals and stamens in the first whole of flowers. So we will look at this in more detail when we encounter the ABC floral patterning, and AP1 is primarily responsible for specifying sepal and petal identity. So we'll see that sepal and petal identity is yeah, for now we learn that petal and sepal identity is by AP1. So mutation in AP1, we can have missing sepals and petals. Mutation in AP1, which identifies sepal and petal, will be gone. And constance is also a transcription factor that plays a central role in controlling timing of flowering in response to day length. So this is another phenomenon in plant. The plant can regulate the timing of its flowering in response to day length. It promotes flowering under long day conditions. So when there is ample sunlight, when the sunlight exposure to the plant is long, and in long days, we have this gene constans as a transcription factor promoting flowering. In Arabidopsis, which is a long day plant, constans is expressed when days are long, and its protein product induces expression of flowering genes like flowering locus T. FT in turn promotes transition from vegetative growth to flowering growth. So then the plants can decide when they switch from vegetative phase to the reproductive phase. And vernalization is just the opposite. Vernalization is when we have shorter days or when we have low temperature, then the plant will freeze. We kind of reserve the plant will preserve the flowering identity or the flowering genes, so that in cold temperatures and in short days the flower does not come up, which decreases its chances of sexual reproduction, because producing a flower and maintaining a reproductive organ is quite costly. So we have such genes in plants. Constance is a transcription factor. So if we have, so constance activates flowering locus T, this is also correct. Apetala mutant missing sepals and petals is also correct, and clavata mutant extra organs produced is also correct. Extra primordia, you can say, leaf, shoots, the number of flowers. So the correct answer is hobbit mutant. Hobbit mutant will lead to improper root meristem because hobbit affects root meristem. Moving on to the next question, which of the following is our false? Which of the following is our false? Option A, mutation of B and C group genes as well as sep will cause all four holes to form only sepals. Mutation of B and C group genes but not sep will cause all four holes to form only sepals. Ectropic expression of sep in leaf causes it to develop into a sepal. Mutation of only sep causes all four holes to form sepals. So these are the four statements. Which of the following are false? Mind you, even I had a difficult time in finding the answer to this, so I'm very not sure of this answer, but I'm happy to discuss. I just want to see what the students are thinking are the correct answer. So this is solely based on the ABC flower patterning, mutation of BC group genes and sepalata. Which of the statement is false?
Second and fourth.
Second and fourth, says Ajay. Okay. Mutation of B and C group genes but not sep will cause all four holes to form only sepals. Do you think this will be a false statement? Mutation of B and C group. Mutation of only sep causes all four holes.
One will express only A and it will be all sepals.
Then that is a true statement, Ajay. That's what I was saying. That mutation of B and C, so A expresses, it will cause all the four holes to form only sepals. That is a true statement. So I think that would not be the answer. So let me reveal
No. I said second and fourth, sir. That is true statement.
Yeah, but the question is which of the following are false?
No, so I said 2 and 4.
So option 2 is mutation of BC but not sep. So let me reveal what I have marked as a correct answer. I have marked option 1 as the correct answer. This is a false statement. All other statements are true.
Okay. As well as.
Yeah, so we have B, C and sep. All the three mutated.
Sep
Here.
means it stands for A.
Yeah, A. So let me just.
English is not good. I
Yeah.
thought
It's okay. It's okay. So yeah. So this is a ABC patterning, the ABC floral patterning diagram. It's pretty simple to understand. I'll simplify it for everyone else further. So we have the A gene, as Ajay and I were discussing. A stands for the sepalata. Sorry. A stands for the gene that is forming the sepals. We have B, C, D and E genes. All these genes, different combinations of these genes will produce a different structure. So sepals is produced when there is only A in the presence of E. So we have this color coding here. A plus E will form a sepal. A plus B plus E will form a petal. Then C plus B plus E will form a stamen. C plus E will form a carpel. C, D and E will form ovule. So this is the pattern in which the floral organization will happen. Now let us see what will happen with mutations in B. So if the B gene is mutated, apetala 3 is mutated. So what is the function of apetala 3? When functioning normally, it is responsible for specifying petal identity. So we have petal identity and stamen identity. So if you remove B, we do not have petals and stamen. So there will be sepal, carpel and ovule. So mutations in the AP3 gene can lead to transformation of petals. Petal will look like sepal and occasionally into carpel-like structures. Because since the stamen is not there, now with C and E it will form the carpel. In extreme cases, mutation can result in complete loss of petals, causing a flower with only sepals and stamen. Sorry, not stamen, sepals and carpel. Sorry for this mistake. I'll just just rectify this. Yes, this transformation of petal identity is often referred to as B class mutant phenotype. So we have B identifying petal and stamen. If we do not have B, no petal, no stamen, everything looks like sepal or carpel. Mutations in C. Now you see this big gene identifying so many things: stamen, carpel, and ovule. If you do not have C, pistillata, which is PI, also known as epitala 3-like, is another gene that specifies stamen carpel identity. PI primarily specifies stamen identity, works together with AP3, means B, to determine characteristics of the stamen. Mutation in the PI gene can result in transformation of stamen into petal-like structures. So now you see, if you do not have C, we have A, B, and E. Now A, B, and E will form a petal-like structure. So the stamen will be absent and it will more look like a petal. And in extreme cases, stamens may be completely converted into petals. So this is what happens in extreme cases of C mutations. This transformation of stamen, often referred to as C class mutant. So we understood B class and C class mutant. Now in the question we had B and C mutation simultaneously. Now if we have no B and no C, then A will prevail everywhere and we'll have everything converted into sepal. Even this will not form the ovule, because to form the ovule we need D, C as well as E. Now there is no B and C, the A will prevail. So in another diagram, if I simplify it for you guys, this is more easier to understand. A and C inhibit each other. So in sepals we have A plus E. So if you see here, A gives these protein, AP1, sep, E gives the sep. Petals, A plus B plus E. So these yellow things come from the B. Stamens, we have C playing here, AG. C here, so we have red thing. Carpels, red and only sep. Ovules, green, D gives the green thing. We have also red and this blue from the E gene. So mutations in B and C only and not sep will lead to all 4 sepals. So only mutation in B and C means there are no yellow and no red. So no yellow, so petals will not form, stamens will not form. No red, so carpel, ovule and stamens will not form. But all the holes will be converted into sepals. So we will have mutation, we will have all 4 sepals when mutation in only B and C and not sep. In case of ectopic sep expression in leaves, if we express this E gene ectopically in leaves, the leaf tissue receives the ectopic expression of sep, may acquire characteristics of sepal, including altered morphology and gene expression patterns. So sep is so powerful, if we ectopically express it in leaf, then it will make the leaf look like a sepal. This transformation occurs because sep genes are involved in specifying identity of sepals in the first floral hole. When sep genes are mutated or non-functional, the genetic program responsible for specifying identity of these organs is disrupted. In a quartet mutant, or a case where all 4 sep genes are mutated, the following transformations can occur. So when all 4 sep genes are gone, sep genes are totally gone, like we have mutation of only sep causes all 4 holes to form sepals. So then this will also be correct. So the correct answer would be mutation of BC group genes as well as sep will call all 4 holes to form only sepals. This will not be correct, because we need the sep to form sepals. And so this is the first statement. Whereas mutation of only one sep causes all 4 holes to form sepal, because there are other seps to form the sepals. We move on to the next question. Which of the following is or false in mammalian development? So now we enter into mammalian development or human development specifically. Which of these are false in mammalian development? Maternal mRNA and proteins in the oocyte are major players in the early stages. Rotational cleavages 1 and 2 are important for cytoplasmic segregation of fate-specifying factors. All cells simultaneously undergo mitosis in early stages. Defects in zona pellucida may cause implantation of the embryo in the fallopian tubes. These are the 4 statements. Please let me know which do you think is the correct answer. Let's take a minute. Which of the following are false statements regarding mammalian development? Okay, I see Ajay has answered option 3. Let's take some more time, and to all the students who will watch this video, read all the statements carefully and think about the correct answer. Okay, it's time for me to reveal which is the correct answer. Which of the following are false in mammalian development? And the answer to this question is option 4. Defects in zona pellucida may cause implantation of the embryo in the fallopian tubes. This is a false statement. All the rest three statements are true statements. Maternal mRNA and proteins, major players in the early stages. Rotational cleavage 1 and 2, important for cytoplasmic segregation of fate-specifying factors, is also a correct statement. All cells undergo simultaneously mitosis in early stages is also a correct statement. Let us see how all these statements are correct. So if you see let's talk about rotational cleavage. Mammalian embryos undergo series of rotational cleavage divisions that lead to formation of a multicellular blastocyst. So blastocyst is a stage where we have the inner cell mass, the trophoblast, and the cavity called blastocele, and a single celled zygote undergoes a series of rotational cleavage divisions to form the multicellular blastocyst. During these divisions, the cells become progressively smaller and referred to as blastomeres. So what happens? The division happens so fast that without increase in cell volume, there is increase in cell number. This type of division is referred to as cleavage, and cells become progressively smaller, referred to as blastomeres. These divisions are primarily symmetrical and result in relatively equal distribution of cytoplasmic factors among the daughter cells. So the daughter cells now have distribution of these cytoplasmic factors because of rotational cleavage divisions in the mammal. And in mammalian development, maternal mRNA and proteins do play important roles, especially during early stage of development. So even before fertilization happens, the mother lays the egg with maternal mRNA and proteins already infused inside the egg. Now when the sperm starts fusing, the maternal mRNAs are dumped into the zygote. These maternal factors are critical for initiating and guiding the development of the embryo before zygotic gene expression becomes the dominant regulatory mechanism. So we have also seen such phenomena in Drosophila, which is an invertebrate. In Drosophila, we still have maternal genes playing a lot of effect. Maternal genes leading to gap genes and controlling pair rule, segment polarity, and other genes. And Hox genes are the zygotic genes which come into play. So here, similarly, in mammals, the maternal gene organize or guide the development of the embryo before zygotic gene expression starts. And for the simultaneous mitotic divisions, which I was talking about, early division of the zygote into multiple cells without increase in size, partition contents. This is known as cleavage. So the mammalian embryo undergoes cleavage divisions where cells undergo synchronized or simultaneous mitosis to generate more number of cells without increase in volume of the cytoplasm. So with similar volume of the cytoplasm, one cells become 2, then 4, 8, 16, 32. So each cell is undergoing simultaneous mitosis. So when we have the two cell stage, each of the cell now divides into two at the same speed and synchronized to form a 4 cell stage, then the morula or the 8 to 16 cell stage, then we have the blastocyst, morula solid ball of cells. So the synchronized division is also a true statement, whereas the fourth statement is not a true statement, because if you recall what is the function of zona pellucida, while the zona pellucida is essential for earlier stages of development, including fertilization and early cleavage, it is not directly involved in implantation. So what happens is zona pellucida is very important for sperm guidance and fertilization. Without zona pellucida, there can be multiple sperms fusing with a single egg, and also competent sperm cannot be competent sperm cannot be filtered out. So the zona pellucida only allows one sperm to fuse with one egg. So it is very important during fertilization. But what happens during implantation after fertilization? There is a process called hatching where the zona pellucida ruptures, and these the blastocyst, now in the blastocyst stage, comes and attaches to the endometrial lining of the uterus with its trophoblast, where we do not have the zona pellucida. So the zona pellucida is not responsible for implantation but very important for fertilization. So hence, if we have any defects in zona pellucida, it might affect the latest earlier stages of fertilization, but it will not affect its implantation. So implantation will happen regularly at the uterus, but the fertilization process will be hampered. Implantation refers to the attachment of blastocyst to the uterine lining, typically occurring within the uterus. So we have zona pellucida hatching, which causes adhesion of the embryo into the endometrial lining, the luminal epithelium, with the trophoectoderm. So the fact trophoectoderm needs to be exposed so as to interact with the endometrial lining. Yes, Ajay, you have something to say.
Suppose zona pellucida hatch before this embryo reach to the uterus, so it will be implanted into the tube. So it will be not directly related with the implantation, but indirectly. That is, zona pellucida is like a cover, no?
Yes,
Unless
yes,
the cover is broken, the blastocyst won't be get implanted.
yes. You're correct.
Suppose the if cover broken early, so it will be implanted into the tube, and that is the reason for tubal pregnancy, or means pregnancy is
Yes, I,
at
I
the
understand. I understand, Ajay. But here,
ectopic. That's what they were called it the ectopic pregnancy, I think.
ectopic presence pregnancy is like apart from the uterus if it implants somewhere else.
Somewhere else. So
Yeah.
so suppose this cover is removed, what the blastocell will do? It will implant where it is. No?
Yes, yes. But actually, the question here, defects in zona pellucida. So it covers a whole lot of things, which can be very confusing. So one kind of defect can also be,
That's what they said. It may cause implantation of embryo in the fallopian tubes. Pellucida remains there till the blastocysts remain in the fallopian tube. That is like a cover. Means you are moving some thing covered with a cover, and that cover, if that is broken up, that will
yes,
stick up there only.
yes, Ajay, yes, yes. I understand what you're trying to say. If the cover breaks up early, then uh,
Place it will not.
wherever it finds the place, it will just implant. Yeah,
I don't know
sounds.
exactly the figure number, but the figure was very well explained in the by Subramaniam sir. In that the zona pellucida was marked in the fallopian tube, just in a second stage.
Okay. Yeah, I need to have a look about it. Then you might be right. Uh, then this is also a true statement. I was thinking the other way around, that if the zona pellucida, if there is a defect in zona pellucida which does not let it break down, so it is always there over the fertilized
Why
embryo.
I mentioned third is key because the development is asynchronous in mammalian. That's not they the cells don't undergo simultaneous mitosis. That is means it is decided by the other cells.
In the early stages, but if you look at in 2 cell to 4 cell and 8 cell stages, each of the cell undergoes mitosis at the same time.
No, no. First thing the mention Subramaniam sir mentioned it is asynchronous. That means all cells in early stages is not going simultaneous mitosis. Let me. Can I have 1 minute to check, sir? Or you
Yes,
also
yes,
check.
yes, yes, yes,
I
I'll
will
also
tell you
check.
exactly figure and what sir has.
Okay.
They say कि from morula stage only the cell differentiation starts. Means as soon as cell differentiation start, that means mitosis is not taking simultaneously.
Yes,
One
yes,
has to
yes.
function as a tropoblast and another has to
Yeah,
function
yeah,
for
that
forming
that
the.
I agree to, because uh, in early stages, I thought early stages here means the 2 cell, 4 cell to 8 cell stages. So
We
then the
can
mitosis has to, the each cell
it is
has
not
to.
clearly defined. Early stage is to be defined at 16th stage or early stage because till the early stage means till the implantation has occurred and heart starts beating at 12 weeks means organ
Okay.
formation is there. All stages are called as early stages. Still, or you can call it till 19 days early stages. We call it as an embryo.
Mm-hmm.
And the 16th cell stage is called medulla, and which is sealed off from the outside. As soon as it is sealed off from the outside, then the hypoblast and epiblast will develop,
Yes.
and that's why that's how the organ formation will be guided by the other thing. It's not pure means. Why I said the answer three is because Sir has clearly said it is asynchronous. It is not means.
Yeah, and after discussion with you, I also think that option 4 is the correct statement. That zona pellucida may lead to implantation in the fallopian tube. Because if fertilization has already happened, which is also in the fallopian tube, and the zona pellucida is disappearing before even reaching to the uterus, then we can have an implantation in the fallopian tube. Yes, I think you are correct.
So it's not the thing of you and me correct. What they think is correct is more important most of the time.
So
Okay, thank you, sir.
no, this is a discussion section. So I might be wrong. So no issues in that. I just have to. So this will be the correct answer because all cells simultaneously undergo mitosis in early stages. Yeah, differentiation. Once the differentiation starts, then we cannot say for sure. So you are correct. Okay, let's move on to the next questions. That which of the following layers does the inner cell mass not give rise to? This could be a pretty simple question. Which of the following layers does the inner cell mass not give rise to? It is not produced by the inner cell mass. Option A, trophoblast. B, epiblast. C, decidua. D, blastocele. And E, extraembryonic mesoderm. Inner cell mass does not give rise to which of these layers? Okay, I see some answers. Ajay has answered option 1, option 3.
Actually, it is 1, 3, and I think no, extra immunity it will give rise to 1, 3, and 4. Blastos.
Okay, let me reveal what is the correct answer. Option 4 is correct. Option 3 is also correct. Option 1 is correct. You are absolutely right. Well done, Ajay. So you have answered it correctly. Trophoblast, decidua, blastocele, all these three structures not formed by the inner cell mass. So if you look at this blastocyst stage, we have the zona pellucida, embryo shell that protects the embryo until implantation, blastocyst cavity or called the blastocele, trophoectoderm which provides embryo with all the essential nutrients until implantation, and it also plays a major role in implantation, forms the placenta and different protecting structures for the fetus. So it forms the extraembryonic tissue. And inner cell mass, the stem cells, forms a fetus.
So see, you had yourself written zona pellucida embryo shell that protects the embryo until implantation.
Yes, yes. So no, I was thinking on the other way around. If zona pellucida is not getting ruptured, so can it implant? So that's why I stroke off the option in the previous question. That may lead to implantation in the fallopian tube. If zona pellucida has a defect which persists the zona pellucida, that it does not degrade, then I thought that it would not implant in the fallopian tube. That's what I was thinking. But yeah, you are also correct. So I had to change my options.
So just
Okay.
my philosophy is question can be confusing, but answer should not be.
Yes, yes.
Whoever had prepared that MCQ, actually these are not MCQs, these are MCAs, multiple choice answers.
Yes, yes.
Whoever they
So
prepared that, he should be clear
you
in
might
the
be
mind, that
facing
will be the
you might be
answer.
facing such questions in your quizzes also. And
Yes, sir.
believe me, it's not me who is designing the MCQs. I'm trying to find out the answers for the MCQs to help the students.
actually, I just given the exam for the proteomics, but
Yeah.
in exam they don't ask this multiple choice answer. They only one was correct. That's they asked. You
Okay.
have to just
Okay.
pinpoint one. This
Okay.
is given
Let's.
for the practice, I think, for understanding purpose of the student.
Hmm.
But what does it give harms? Internal assessment marks, they are always lower.
Oh,
Means
okay. Yeah, that
mean I got
completely depends on
97% in
the.
the last, and my total because of internal assessment it reduced to 94%. Total percentage was 87% in RNA biology, and then it reduced to 84% because of the internal assessment.
Okay. Okay. No, you need not be disheartened. You are already doing very well, and all the best. Okay. So let's
Thank you.
move on with our explanation. And here is a implanted embryo with all its extra embryonic structures. So we have the placenta, the myometrium, endometrium, myometrium are the uterine linings. Decidua is the is also an extra embryonic tissue. We have the decidua basalis, basal decidua, parietal decidua, amniotic cavity, and all other structures. So decidua is also an extra embryonic tissue, and the extra embryonic tissue comes from the trophoectoderm. So inner cell mass cannot give rise to extra embryonic tissue like placental structures. So all the structures with placenta or the trophoblast and the blastocele. So blastocele is the cavity, so it does not give rise to the cavity. Inner cell mass will not give rise to the cavity. Also the trophoectoderm or the trophoblast will not be formed by the ICM and the placental structures. So Ajay was absolutely correct, and I hope other students who will watching this video will able to figure out. Moving on to the ninth question, match the following. On the left hand side we have transcription factors, and on the right hand side we have the features of the transcription factor. Nanog, GATA6, CDX2, STAT3, YAP. Then on the right hand side we have all cells express this at morula stage, required for self renewal of inner cell mass, specific to the hypoblast, activated by TEAD4, prevents hypoblast formation. And the options are A2, B3, C1, D5, E4, A5, B3, C14, D2, A4, A5, B3, D2, E14. Option D, A1, B5, C2, D4, E3. So you have to carefully match the transcription factor with its feature. Let's wait for a minute and check the answers. Okay, so let me reveal the correct answer to this question. Option 2 is the correct answer. A5, nanog to prevent hypoblast formation. GATA6, specific to hypoblast. CDX2, all cells express this at the morula stage. STAT3, also CDX2 will match, activated by TEAD4. STAT3, required for self renewal of inner cell mass. So Ajay was correct in A, B, and D, but uh, option E and option C, YAP and CDX2, just very closely related. YAP leads to activation of TEAD4, and TEAD4 then leads to activation of CDX2. So this is the relationship that I was talking about. We have all these four transcription factors, four Yamanaka factors: SOX2, NANOG, OCT4, three of them are Yamanaka, and CDX2. So there are many things that these people regulate at the ICM and the trophoectoderm: epiblast segregation, pluripotency maintenance, cell proliferation, and YAP signaling. YAP leads to TEAD4 activation, leads to CDX2 activation. So let us visit these transcription factors one by one. NANOG is a transcription factor involved in maintaining pluripotency in the embryonic stem cells. It plays a key role in preventing these cells from differentiating into the hypoblast cell types too early. So it maintains stemness, it prevents hypoblast differentiation. So first option would be prevents hypoblast differentiation. Correct. GATA6. GATA6 is a transcription factor crucial for specifying maintaining identity of the hypoblast. GATA6 is just opposing the NANOG. It is expressed in the trophoectoderm, one of the two cell lineages that form during blastocyst development. So trophoectoderm has GATA6 expression and maintains the identity of hypoblast. GATA6 specific to hypoblast. CDX2. CDX2 is expressed by all cells in the morula stage. So this is a gene transcription factor expressed in all cells in the morula stage, but as the stage progresses, it is restricted to the trophoectoderm. It is activated by TEAD4 via YAP signaling. It is a key regulator of the trophoectoderm, one of the two primary cell lineages that emerge during blastocyst. CDX2 also plays a role in establishing polarity within the blastocyst, ensuring that trophoectoderm remains on the outside, where it will eventually facilitate implantation into the uterine wall. So CDX2 will ensure that trophoectoderm is on the outside and the inner cell mass in the inside. And CDX2 is uniformly expressed in morula stage, but then later as the stage progresses, the expression is restricted. STAT3. STAT stands for signal transducer and activator of transcription factor 3, is also a transcription factor that can be activated by various signaling pathways such as JAK-STAT pathway. In the context of embryonic development, STAT3 activation is involved in maintenance of pluripotency in the inner cell mass. So STAT3 will go for inner cell mass, CDX2 for all cells in morula expression, and later in the trophoectoderm, activation by TEAD4, GATA6 for hypoblast, NANOG for preventing hypoblast. So this is the correct match. In return, YAP activates TEAD4, TEAD4 activates CDX2, and CDX2 is expressed in all cells at the morula stage. So YAP is the upstream player in the signaling, which activates TEAD4, then leading to CDX2. So this is what we're talking about: YAP1, TEAD4, then CDX2. And CDX2 can specify the trophoectoderm. And the STAT3 and SOX2 transcription factors can specify the inner cell mass. NANOG will also prevent hypoblast differentiation too early and maintenance of the stem cell. Coming on to the last question of this session. First attachment of the embryo to the uterine wall is mediated by dash on the embryo and dash on the uterine wall. The very first attachment options are L-selectin, sulfated polysaccharides, L-selectin, integrins and actin, collagen and integrins. The first attachment of the embryo to the uterine wall is mediated by dash on the embryo and dash on the uterine wall. So there are two components. We answer this and we finish our practice problem session for this week. I see Ajay has answered option 1, and I had given enough time for all the viewers to think about the answer. And option 1 is the correct answer. Well done, Ajay. L-selectin on the embryo and sulfated polysaccharides on the uterine wall is the first attachment site of the embryo to the uterine wall. L-selectins and sulfated polysaccharides both play important roles in the process of implantation, which is the attachment of the embryo to the uterine wall. So here is a schematic showing easy representation of implantation of the embryo into the endometrial lining or the uterine wall. So in the uterine cavity, we have endometrial epithelium, and some of these glands are also there. In the blastocyst stage, the empty hollow space, fluid filled space, is the blastocele cavity, and we have the embryoblast. Embryoblast here refers to the inner cell mass, and on the outer side we have the trophoectoderm. So trophoblast, which constitutes of cytotrophoblast, syncytiotrophoblast. So this is when all this embryo is penetrating inside the endometrial lining, and at later stages, when the embryo is embedded, new endometrial lining is formed. Hypoblast, epiblast. So inner cell mass then differentiates into hypoblast, epiblast. So this is a schematic showing 3 stages of implantation of the embryo. So let's come back to L-selectins and sulfated polysaccharides. These molecules are involved in initial adhesion and recognition events that occur between the trophoblast and the uterine lining. So the uterine lining has the extracellular matrix rich in sulfated polysaccharides, which are negatively charged polysaccharides, which can bind to certain components expressed on the trophoectoderm. L-selectins are also ligands which are expressed in the trophoectoderm, which have receptors in the uterine wall, which helps in attachment of the embryo into the endometrial lining. So here is again a representation of different players in the implantation process. Implantation takes place with the apposition of the blastocyst at the endometrial epithelium. Blastocyst cannot be displaced anymore. So when the blastocyst is flowing from the fallopian tube towards the uterus, apposition is a process in which this gets oriented and fixed. This is the endometrial lining. Then these are the players that we see here. Blastocyst adheres to endometrial epithelium, establish a stable connection mediated by adhesion molecules, pinopodes and signaling factors. So one of these is L-selectin and its receptors. The other one is sulfated polysaccharides present in the extracellular matrix of the endometrial lining. Trophoectodermic cells invade the epithelial layer until the underlying stroma. Production of proteases facilitates the process and enables nutrition from maternal vascular system. So the trophoectoderm secretes proteases which digest the endometrial lining and helps in penetration of the embryo inside the uterine wall. And then these proteases also rupture some of the these proteases also digest some of the maternal vascular system and derive nutrition. So that forms the nutrition for the embryo. And later, the trophoectoderm will form the placenta, which is a temporary extraembryonic structure formed by the conjugation of both the tissues of mother and the embryo, which facilitates nutrient and blood circulation through the fetus. So we have apposition, adhesion, invasion by the trophoectoderm, secreting proteases, and full implantation. So these are brief phases of implantation of the embryo into the endometrial lining. So L-selectin on the embryo and sulfated polysaccharides on the uterine wall are responsible for this implantation. So with this, we have finished 10 questions corresponding to lecture week 10, and I hope we had a pretty good and fruitful discussion, and all the viewers could derive some knowledge from it. Yeah, I'll just stop presenting my screen. Okay, so since we have come to the end of the presentation, I'll end this call. See you soon next Saturday, where we have 2 weeks more of this problem solving session, and we'll be done covering all the lectures. And I can tell you will be most thoroughly prepared and most thoroughly prepared for the examinations on the October end. All the best to you guys, and keep attending the lectures. And whoever is not able to, you can view these recording sessions which I have prepared for you guys. All the best, and thank you, Ajay. Okay, see you next Saturday. Bye bye.